So 17.3 talks about something I've actually already discussed in here. Really. So it's how they figure out the order of bases on a chromosome. How they sequence the DNA sequencing. And when you say DNA sequencing, they can take your DNA and figure out, okay, the first letter on the first chromosome is T, the first base. And then C, and then A, and then A, and then T, then G, and then C, and then G. 3 billion letters long. They can sequence your whole genome. They first did this in what was called the Human Genome Project. And it was started when I was about your age, in the late 80s. And it took them like 13 years to do it. And they finally did it. And they had sequenced the whole DNA in a human. Now they can do it in just a matter of a few days. And the method for doing it that was first figured out is called Sanger sequencing. Y'all remember this?
Yeah.
Let me go back over it, even if you do remember it. I mean, not everyone does. It uses these DDNTPs. Come on, remote control mouse. Dideoxonucleotides. Can you say dideoxonucleotides?
Dideoxonucleotides.
Pretty good. And we call these DDNTPs. And so, why do we call it dideoxo? Because the normal... Do you remember that a normal nucleotide in DNA has the sugar? What's the name of the sugar in DNA? The deoxyribose, right? They call it deoxyribose because it's basically a ribose sugar that's missing an oxygen. Deoxy. So why do you think they would call this dideoxy? It's missing 2 oxygens. That's why it's dideoxy. Where's the other oxygen that it's missing? Do you see here's a normal deoxyribose sugar, and there, see how it has OH on the bottom? You'll notice that the dideoxy is missing that O there. It's just got H. You'll see the difference? Well, here's the thing. When you're missing that little OH group, and you just have an H there, you cannot attach another nucleotide to the bottom like that. It needs that OH group. Do you remember when you're attaching 2 things together to make a covalent bond, then an OH comes off one side and an H comes off the other to make H2O? Y'all remember that? What kind of reaction is that when we take OH and H and make H2O?
Dehydration.
The dehydration reaction. Do it, Josie. It's also called a condensation reaction. Anyway, you take away an OH and an H, and you can put 2 things together. Well, if you're missing that OH, like this one is, you can't hook anything to it. So scientists can actually make, in the lab, these dideoxonucleotides. And they make them so that when you shine a laser at them, they fluoresce in different colors. The adenine DDNTP fluoresces green. The cytosine DDNTP fluoresces purple. The thymine DDNTP fluoresces red. And the guanine DDNTP fluoresces yellow. Yes? Oh my gosh, you're going to miss all this. What a terrible time to go. Yes. Go ahead. Okay, so how does this work? Remember, these cannot have any new nucleotides attached to the bottom of them. So basically, these DDNTPs are going to shut off DNA replication. So what do? If you want to figure out the letters of this gene, you have basically a machine that heats up the DNA real hot. What do you think will happen to the DNA if we heat it up real hot?
DNA trees.
It'll separate and come apart. And then, wait a sec. Let's do it like this. Works better with my models if I have it that way. Let me give myself some room. Have too many magnets. Magnet mania. All right, here we go. Heat the DNA up, it separates. Let's just get rid of this side. And let's try to figure out the sequence on this side. Y'all understand if we know the sequence on this side, we can figure out the sequence on the other side, right?
Yeah.
Because there's rules about how they pair.
Right.
Matt, you with me? All right. You also put a bunch of free normal nucleotides into the mix. You let it cool down. You also put these little DNA, these are called DNA primers. And they have a certain known sequence. So to be able to sequence the gene, you have to know the beginning sequence. In this case, we know that the first three are TAC. Now, how scientists figure that out is in another way. But anyway, we know that the first sequence is TAC. We can make an ATG primer. This ATG primer will stick to TAC. And that can get the process started. Now, in actuality, the primers are longer than just 3 bases. They might be like 15 bases long. Okay? But what will happen is you let it cool down, you heat it so it separates, you let it cool down. These aren't able to stick, but a primer can stick to the beginning. Now, if you put in a DNA polymerase enzyme, also in your test tube, the DNA polymerase can start adding new nucleotides after the primer. And that's what it'll do. It'll grab a nucleotide and add it to the end of the primer. Always adding in the 5' to 3' direction. Take a nucleotide, add it to the end of the primer. Take a nucleotide, add it to the end of the primer. Now, at some point, it'll just randomly grab one of these DDNTPs that you have in the mix. So here we go. A G is next, so it's going to attach a C to it. But let's say it grabs this DDNTP and attaches it. Now, it cannot grab another nucleotide and attach it after that one, because this one is missing that OH group, right? So we have, in effect, terminated the sequence. That's why they call these DDNTPs terminator bases. Y'all remember The Terminator? Great movie with Arnold Schwarzenegger? Totally unrelated to this, but very good. So, the sequence is terminated. The DNA will be heated up again and will tear apart. And this little sequence right here just floats away. I'm going to set it to the side over here. It's just floating around in the test tube. And you just do it all again. Okay? And you're going to do this over and over and over. So let's do it again. Primer, attach it to the sequence. DNA polymerase grabs nucleotides, puts one down, and it grabbed a DDNTP just by random chance, puts one in. Heat it up again. This breaks. Now we have that one. And you'll just keep doing this. Let's do one more. That attaches. Now the DNA polymerase can add nucleotides. Let's say right away, let's say the first one it grabs is a DDNTP. You got to put the right numbers of DDNTPs there so it will randomly stop it enough. And you can see we have different length partial strands that are coming off of this every time we do it. You know what? I put the wrong one, y'all. Why did somebody say something?
You just say something to fall off every time it happens?
So every time you heat it up, this thing just falls off of that one, and now you have different length strands. Yes?
Two questions. One,
Yes.
Can it be any letter, like the nucleotides? Can they be like any?
Any of the four, A, T, C, G.
Okay. And two, can I go to the bathroom?
Yes, but you're going to miss the grand finale.
Oh, it's okay. I'll learn it.
Okay. Y'all need to pick better times to go to the bathroom. All right. Hold on. Let's make one more. Probe kicks into place. DNA polymerase adds one there. And it picked up a DDNTP, adds one there. This breaks off. So now we have, after doing that over and over, you will have some fragments that are short, just one base after the primer. And you'll have some fragments that are longer, maybe 4 bases after the primer. So they're different lengths. Yes?
Is the primer the same sequence for this?
The primer is always the beginning sequence. So it's always, for my example here, the primer is always ATG, because that matches the beginning.
Okay.
So the primer will always attach to the beginning of the gene that you're wanting to sequence. So all you got to do if you're the scientist is come up with the primer, and then you can do this Sanger sequencing thing.
And then they're already cut up into sequence, like in the test tube, they're already cut up into specific genes.
What are already cut up?
To where they're
When you say they are already cut up, what are you talking about?
the long strand.
The DNA? Yeah, you can cut it up into pieces.
And then they find primers for each.
For each piece, that's right. You got to get a primer for each piece.
Multiple of those DNAs in one test tube, or they try to get just one DNA?
That's a good question. It's a good question. I think they separate out the pieces. This is actually done in like a container that has like thousands of little wells in it. And I actually think all the DNA is in each one, but you have only the primers for the section you're interested in. That's how it works. I've never actually done this in the lab. So, now that we have all these different sized pieces, you take take a pipette. I'll make it go across like this. You take a pipette, a tube, that's filled with agar. Y'all know what agar is?
The stuff in the petri
The
dish.
thick stuff in the petri dishes. It's viscous. Have you ever heard that word?
Yeah.
Viscous means thick, like oil. It's not like water. It's thick. And you actually, I like to say you suction these up, but what's actually happening is you're applying an electric current. You have a positive charge over here and a negative charge over here. And that'll push these pieces up into the pipette, you see. Oops. And the pieces get pushed up into the pipette. Now the pieces will separate themselves through the agar. The light pieces, the smaller pieces, will move fast through the agar. The longer pieces will move slow through the agar. So they'll line up through the agar like this. Wait, this is the longest piece. Let me put that in front.
Do you think there's ever air?
There is, but I'll show you how to correct for that. So in actuality, there's not just one little piece. There's thousands of little pieces that are 4 nucleotides long. There's thousands of little pieces that are 5 nucleotides long. There's thousands of little pieces that are 6 nucleotides long. And they're all coming through this thing. And what they do is they put a laser right here. Here's the laser. Laser, which shoots a little laser gun shoots a little laser light through the agar right there. Dang it, that thing keeps falling. And so, as this thing's coming through, and it passes the laser beam, it will glow green. And green means A. So A comes first. Now there's not just one of these passing by, there's thousands of them. So, and they're all A's, because A was the first nucleotide after the primer. And that's why you get this hump right here. This shows all of this. There's a hump showing how many G's. For this picture, they have G as black. G actually fluoresces yellow. I don't know why they didn't use yellow for G. Terribly written. Anyway, that's why you see the hump, Josie, there, because we had just a peak of G's come through. And then there was a short time period. I mean, this is milliseconds. And then you have a whole bunch of A's come through. In my example here, A comes through first, and then C comes through second. So C is the second one. And then in my example, G comes through third. So G is the third one. And then C comes through fourth. So C is the fourth one. So, as these things are coming through, you're reading the sequence. And so we know the primer was ATG, and then we know the next base, and the next base, and the next base, and the next base. You just read these off so the computer can build a sequence. Now, Thomas brought up a good point. They don't do the whole gene. The whole gene is like 100 million letters long for one chromosome. They cut it into pieces. So this is a smaller piece, and let's say it reads ACGC, AACA, GTAC. And that's the first gene that they cut. They'll have another piece somewhere else that goes AGTAC, AGATACCAGA. And what they'll figure out, what the computer can figure out is, since this AGTAC matches this AGTAC, this piece and that piece like overlap. It's like putting a puzzle together almost. The computer can figure out, oh, we have overlapping sequences. This must be the next part of the gene. So it must go like this and then there. And the computer can put all these things together, you see, and get the whole chromosome. So you start by cutting the gene into thousands of pieces and putting all of those into these little wells. These little wells, it looks like a... I'll show you what it looks like. We got them in here. Have you ever used these in chemistry?
The flush, the camera stools.
You ever use these things?
We use some like more.
Can't say that I have.
This is for doing a whole bunch of reactions. Each little well, you cut the DNA to pieces and you put them in each one. Now imagine this, where the little wells are a lot smaller and this grid is a lot bigger. You can do thousands at a time, you see, and run each of them through.
Does that keep it from missing a nucleotide?
Yeah, because you do so much, you're going to have, like I say, each of these little peaks you see is thousands of pieces that are of a certain length. You know, you don't just have one piece going through, you have thousands. So as they sort themselves by size, you're not going to miss anything because you've done it so many times. And again, your gene is 100 million. One chromosome is 100 million nucleotides long, so you cut it into a lot of pieces. You have to have a different primer, and you put these primers in different wells, and you can eventually piece the whole thing together. What do y'all think? You want to see video footage? Sanger sequencing, baby.
Like actual video footage.
No, I think it's cartoon video footage. You don't like cartoon video footage?
I want to see the video.
Maybe we can find that. This is one of my favorite video lessons. You can see that at all. Yeah, but this one really is.
DNA sequencing is the process of working out the order of the building blocks, or bases, in a strand of DNA. Before we can sequence the DNA, it has to be cut up into smaller pieces that are inserted into plasmid DNA and then put into bacterial cells. This makes it possible to produce lots and lots of copies of it as the bacterial cells multiply. The DNA is then isolated from the bacteria and sent for sequencing. The isolated DNA is transferred to a plate where the sequencing reaction will take place. A mixture of ingredients is added. These include free DNA bases A, C, G, T, DNA polymerase enzyme, and DNA primers. Modified DNA bases labeled with colored, fluorescent tags are also added. These are called terminator bases.
How was watching the video?
To start the sequencing reaction, everything is heated to 96 degrees Celsius. This separates the DNA into 2 single strands. The temperature is then lowered to 50 degrees. This enables the DNA primers to bind to the plasmid DNA. The temperature is then increased to 60 degrees and the enzyme DNA polymerase binds to the primer DNA. DNA polymerase starts making a new strand of DNA by adding unlabeled DNA bases to the target DNA. It continues to add DNA bases until a terminator base is added. These terminator bases have been chemically altered so that no more bases can be added to the new strand of DNA. Once a terminator base is added, the DNA polymerase enzyme stops making DNA and falls away from the strand. Everything is then heated to 96 degrees Celsius again to separate the new DNA strand from the original strand. This process of heating and cooling is repeated again and again to produce lots of fragments of DNA of different lengths. The length of each fragment depends on when a terminator base got added. To read the sequence of the DNA, the various fragments are separated by length using a process called electrophoresis. A capillary tube is lowered into each well of the plate and an electrical charge is applied. This causes the negatively charged DNA molecules to move through the capillary tube. Each capillary contains a porous gel. The shorter fragments of DNA move through the gel more easily than the longer DNA fragments. As a result, the fragments become arranged by size from the shortest to the longest. As the DNA fragments come to the end of the capillary, a laser makes the terminator bases light up. The color is detected by a camera and recorded. Each terminator base is labeled with a different color. A fluoresces green, C blue, G yellow, and T red. The shortest DNA fragments will be read first and the longest read last. The sequencing machine records the color of the terminator bases as a series of colored blocks. Each colored block represents the labeled terminator base at the end of each fragment of DNA. By converting the colors into letters, we get the sequence of our piece of DNA.
Does that make sense? So, it's kind of interesting. You're not actually getting the sequence of this side. You're getting the sequence of this side. It doesn't matter. All you need is one side. Then you can match the other side. And you got the whole sequence of DNA. And that's how they know your code. And they've sequenced, like, most organisms. Let me show you. There's a giant database that has all these organisms in it. If you have your computer, you can find it too. The name of it slipped my mind. Think, Mr. Willis, think. It's, woo! Can't remember the name of this thing. Blast! Ha ha! You just gotta think, see? When you're young, you can think quickly. When you're old, not so good. BLAST. Type in BLAST. BLAST stands for Basic Local Alignment Search Tool. Are we going to have a BLAST? Yeah, we can do this in the lab. Yeah. Click Nucleotide BLAST. You can enter any sequence of DNA into this thing right here. So let me just make up a sequence. A, T, G, C, C, A, A, G, T, C, A, A, G, T, A, A, C, C, A, G, T, A, C, A, A, A. Now, do you think that appears in any organism's DNA anywhere?
Yes.
So, I'm going to go down here to the bottom and I'm going to click BLAST. It's amazing that you can just do this. Any person can search their database. And what it's doing right now, it says searching. It is searching for that sequence in their database to see if it exists in any organism that they've sequenced. And they've sequenced thousands of genes. And sometimes it takes a while. Here's the organisms. See where it says per ident? This is the percentage that it matched. So in every one that says 100, it was a perfect match in these organisms. What is this organism? Dromaeus novalandia. Nuclear receptor. What is Dromaeus novalandia? Well, I can click on here. There's all sorts of things. I can click Genbank and see what this organism is. It is an emu.
Yo.
I clicked, that was a match for the emu DNA. And if you want to see the gene, there's the sequence.
I just thought it landed on.
Mine was a Pacific oyster.
Look at that, look at that. This is just one gene. This is one of its genes.
That's one thing.
This is one gene. Look at all the A's and T's and C's and G's. So that sequence I inputted is in here somewhere. I'm not exactly sure where.
Can we find it?
Yeah, I could search, but I'd have to go back. I don't know how I go back and find out what sequence I put in. Yeah, I could have copied that and then searched. You're right. But anyway, wow, there you go. That's a gene, and they have sequenced this. And you know what these letters stand for right here? Can anyone guess? I'll give you a hint. At the beginning of it says translation.
Oh, was it amino acids?
These are the amino acids. Every amino acid has its own letter. So what do you think M stands for? It's at the very beginning of the gene. Methionine. Do you remember M is the start codon? So that's why the M is first. Because the methionine comes first. Isn't that cool? So these are all the amino acids. Remember when I said a protein is hundreds of amino acids long? This is a big protein. So this shows you the protein, and this shows you the sequence of building that protein.